Okay, so pleasant good afternoon, everyone. And today we'll be doing a bit of a revision session to see what's in line or in store for us as it relates to the spotter next class day. Thus far, what have we covered in terms of materials? Well, we have looked at the blood, heart and blood vessels, the respiratory system, and the digestive system. So in terms of the spotter, material from these things will be present for your spotter itself. So let's see if we could get into a little more specifics as to what maybe you should target your revision on as it relates to these topics. Well, it could be found from the lecture notes, the lab notes, and I'll just point out certain things which are important. When we think about the heart, of course, what's the first thing that comes to mind? I hear what you're saying. So the blood vessels associated with the heart itself. While that may be so, we'll see what exactly you should put emphasis on. I'm just waiting for the PowerPoint to come up. So as soon as it comes up, I'll take it from there. Okay, it's taking its time to come up, so let me talk around it. Heart and blood vessels. So it comes up, it's the blood. Okay, so with the blood, what is important for the blood, and in fact, let me just see something in the chat there. Okay, good afternoon. Yes, Tonya, good afternoon. Okay, so with the blood, you should be able to identify them, the different types of types of blood. So erythrocytes, red blood cells, leukocytes, the white blood cells, and also platelets. So if you're given, let's say, a slide, you should be able to pick out and also particularly differentiate among the different white blood types. So let's have a look to see what do I mean by that. So for this entire, when you're looking at the spotter itself, granted there might be few multiple choice questions. And when I say few, I'm referring in the vicinity of at most 10 or less. The majority of the questions will be based on images. So when you're going over in terms of your revision, you'll be well placed to focus on the images particularly associated with the topic. And I'll highlight them as we go along. Open sesame. All right, so with the white blood cells, there are 5 major types of white blood cells. So you should be able to identify them. And if given that, let's and also pick out a red blood cell, what it looks like, the biconcave shape of the red blood cell, and of course, just the broken up appearance of platelets. In terms of blood vessels, you should be able to pick up or identify vein and a smaller portion of venule, arteries, arterioles, and capillaries. Okay, great. Well, that's 12 come up now. Okay, I wanted 11. There we go. Thank you. On the topic of blood and blood vessels, so when you're going through, all right. So this image with all the different types here. So a neutrophil, in terms of its appearance, right? Know the multi-lobed appearance of the neutrophil. This is an H&E stain. In terms of the pink, you might be wondering, well, hello, what type of blood cells are in the background here by chance? Which ones do you think these are? If you had to make a guess.
The red blood cells.
Where you go. Right, indeed. So they are red. But the reason why they don't appear red is because you have a stain. There's a stain that they use called an H&E stain. Hematoxylin and eosin. It's a kind of dye that they use, and it dyes it pink, the red blood cells. So that's why they appear like this. And the reason why they use it is really to bring out the colors of the white blood cells. So here you see a neutrophil, right? So if given, let's say, an image similar to this and say, you know, what cell is this? You should be able to identify it. Another thing as well, as it relates to the spotter, you wouldn't be asked to write anything or type in anything. Everything is either matching, even though that doesn't work on phones, so I doubt you'll have it, but it would be that you have to select from a group of answers, which one is the correct one. The drop-down menu, and you click on it, that would be in force here. So you wouldn't be expected to type anything. So just take note of that. All right. Here we are seeing an eosinophil, and difference with the eosinophil, it dyes pink or reddish in terms of the dye, and then you have this bilobed appearance that makes it look like it's wearing glasses. All right, so these are the eosinophils. This type here, these are the basophils. And notice how the nucleus almost completely fills, as shown here, these two different dye, it almost completely fills the circumference of the cell itself. So basophils usually have a very large nucleus, as opposed to the eosinophil, which you're looking like, it has a bilobe, which makes it look like it has glasses on. Yeah. Okay. So those are the granulocytes, and they're called granulocytes because they have a grainy appearance. So when you're reviewing it, do take note that the white blood cells, they have two types, granulocytes and agranulocytes. Granulocytes, they have a grainy appearance, whereas the agranulocytes, if you notice, the cytoplasm is smooth. It has a smooth appearance. There are no grains present. So specifically, let's have a look. Lymphocyte, oops. Let me take a step back. Lymphocyte, so this is a lymphocyte. This is the nucleus almost filling the entire cell. Here you have a monocyte. And again, it's very similar, if you would. It's similar to the eosinophil, but the eosinophil is grainy. Whereas with the monocyte, yes, you have a similar shaped nucleus, but there are no grains associated with the cytoplasm. It doesn't have a grainy appearance. The next one is the thrombocyte. And the thrombocytes, these are just, these are the platelets, and they are associated with hemostasis or the forming of clots. Now, clots is very important. What would happen if we did not clot, if blood did not clot, or the clotting mechanism was not activated? What would we have? Something that begins with H. When you bleed internally, it's known as hemorrhaging. Very good. Yeah, well done. All right. So do take note. So that's the differences there. Artery and vein, if you're given this image and you're asked, to differentiate between the two, which one is the artery, which one is the vein, you should be able to tell the difference between them. With this one, if you're given, similarly with this one, you should be able to tell, okay, well, what type of blood vessel we're looking at, and it's a capillary. Now, how would you know it's a capillary? Anybody wants to hazard a guess? How do we know there's what are these things that are in the capillary? It's a type of blood cell. Which type of blood cell? RBC, and we're not talking about Royal Bank of Canada. Stands for the RBC, stands for?
Red blood cell.
Thank you. Red blood cell. So if you notice, it just really could accommodate one blood cell at a time. And therefore it's very small. It's approximately about on the order of micrometers. We mentioned about a micrometer. What's a micrometer? I'll just revisit that. If you take your thumb and forefinger, squeeze it together, now pull it apart. You have a little space between your thumb and your forefinger. Divide that little space into 1,000, that is a micrometer. This is about 30 of those. So that's just to tell you how small, it's just incredible that, you know, your body has millions of these capillaries and it circulates the blood all over the place, right? But just to give you an idea how thin this is really thin. And I think now we can appreciate, let's say with some persons, you know, when you do get a bruise, how easy it is for them to break in terms of getting a bruise. And then you see, you know, the skin discoloration looking purplish. It's very easy for these things to be because they're so fine. All right. Aorta wouldn't be required. And neither the systemic, the naming nomenclature of the arteries and veins, no, wouldn't be required for this one. Right, okay, so that's the first one. Let's look at another one. Let's look at the heart, which is number 12. Lab number 12. All right, so for lab number 12, we just looked at the arteries and veins. So the heart, you should appreciate the electrical conductivity associated with the heart. So which one has it? Well, and if we have it here, yeah, right down here. So what we have in the right atrium, we have the SA node. SA node goes to the AV node, so-called because it's between the atria and the ventricle. So this is the atrioventricular node. So SA node to the AV node. Usually around here is the bundle of His. And then the bundle of His, you have your right and left bundle branches. And then that goes down to the level of the Purkinje fibers. So this is how the electrical signal generated at the level of the SA node passes through the entire heart. All right, so do take note. You should be able to label those things which I mentioned, SA node, AV node, bundle of His. Let's see if you could bring up one that shows the location of the bundle of His. Okay. Right. Right, so the SA node, AV node, so the bundle of His is just superior to the bifurcation of the left and right bundle branch. The right bundle branch, the left bundle branch, right? So, but just superior to there, you'll have your bundle of His. So do take note, SA node, AV node, the area superior to that split, that's the bundle of His. And then of course, the little fibers that come off, these would be the Purkinje fibers. Let's look at it in another diagram. SA node, AV node, bundle of His, which is just above the split. You have the bundle of His, you have the right and left bundle branches, and these fibers, fine fibers, looking like roots, these are the Purkinje fibers. Let's look at it in another one. SA node, AV node, bundle of His. This one doesn't show the bundle of His, no. But we will know anatomically it's superior to that little bifurcation or split. So it usually is right there. This is your right and left bundle branches. And then of course the Purkinje fibers, not really shown here, but it's the little roots that come off from this side. Let's look at another diagram. All right, SA node, AV node, bundle of His. also known as the atrioventricular bundle. Right, there's the right and left bundle branches, and these are the Purkinje fibers shown. Usually these are labeled as the Purkinje fibers. Go to like this diagram. Let's get another one. Last one we'll be looking at. Okay. What is the same one? Right. SA node, AV node, bundle of His. Left and right, right and left bundle branches, Purkinje fibers. Okay, good. Let's get back to our PowerPoint. So you should be able to learn, label that as it relates to those things. Type of tissue, cardiac tissue, if given an image of cardiac tissue, should be able to identify it. Now, mark you, it does look similar to smooth spoon muscle tissue. They do have a similarity. Show you what I mean.
Hi, sir. Oh, God.
Yes, hello.
Yes, Tamara is waiting to come in.
Mm-hmm. Yeah, thank you. Right, so smooth muscle tissue and cardiac tissue. Right, so if we were to put them, sometimes they could tend to look very similar. I don't know if this is a schematic. This is more of a cartoon. Let's see if we could get the real tissue. It's more of a cartoon as well. Okay. Okay, yeah, this one's good. So if you were to look at the different types of muscle tissue, you have cardiac, you have smooth, and then you have skeletal muscle. I'm not getting it. More bulge up. See? Right? So what we would notice here. Okay? But the skeletal muscle is not showing up too clearly here. I don't like this diagram. The cardiac muscle, they have these parallel or perpendicular lines, perpendicular to the grain of the muscle, and they are known as intercalated discs. And that's how you can tell the difference, because sometimes you could confuse skeletal with cardiac muscle. They tend to look similar, but the one way of telling the difference, the surefire way, is to look for those intercalated discs. And let's see, this one looks a little better. Yeah, this one is better. Let's have a look. Right, so this here is not showing up clear. You see these lines that usually come down, and they're usually quite prominent. I don't like it. Okay. Oh yeah. Oh, I guess this will work. Usually, right. So the tissue, this is cardiac. Not seeing it here. Yeah. Well, so if you're, I don't know if it's showing up, it's not too clear. You see there are lines that come down. Let me do this to make it even simpler. Right, for you to observe them. Right, this is a lot better. So when you're looking at cardiac muscle, you see these lines, they come down, and that's how you can tell the difference. They are known as intercalated discs. The intercalated disc, what they do, they are really extensions of the electrical conductive system associated with the heart. So while we do have that extension or the initiation of the beat of the electrical electrical impulse that causes the heart to contract, is generated at the SA node, then to the AV node, AV bundle or bundle of His, left and right bundle branches, then it goes off into the Purkinje fibers. After it passes through from the Purkinje fibers to the tissue itself, it will pass along these intercalated discs. So what the intercalated discs do, they help spread that electrical impulse to the fibers themselves. Of course, when they get it, they then contract, and that's how you get the motion associated with the heart, the beat. You have the contraction and relaxation. So the intercalated discs, they assist in getting that electrical impulse generated at the SA node to all the different individual fibers associated with the muscle fiber or cardiac fiber, muscle fibers within the heart itself. Let's get back to our point. Good. So do take note of that. If you see this image, from the time you see those lines, you know those are intercalated discs. Right? No. Okay, so we're looking at the heart. What is this here, this leafy thing? Now, of course, this is a cadaver, which is a preserved specimen. They traditionally use formaldehyde as a preservative. I think they have some newer ones now. But this is a preserved specimen. That's why it has this gray appearance. What is this covering on the heart called? Anybody?
So could you repeat what you asked?
Sure. What is this covering on the heart known as? Give you a hint, it's the peri. What is the word associated with heart?
Pericardium.
Yeah, right over here, pericardium. So this is actually this waxy coating is the pericardium. And what does the pericardium do? What does it prevent? One word begins with F. Pardon? Fuss? Sorry, I'm not hearing you too clear.
Fat.
Or fat. Or if it is fat, it does have fat associated with the heart, yes. And in fact, let me think. Yeah, what we're looking at here, no, you're quite right. Now that I look more closely, this is the fat. This is fat around the heart, but this is the pericardium out here. So you're quite right. This is fat, right? And the fat serves both as a cushioning, and it also aids in the prevention. And what does it prevent?
Internet unstable, so it keeps dropping.
Right. So what it prevents is it prevents. Right. So what does it? It prevents this pericardium. It prevents what from occurring between the heart and the lung. So you have to remember this is in the mediastinum. This here coming down here. This will be the esophagus in the back and the trachea to the front. So the trachea then bifurcates to the left and right lung. And then, so the pericardium, it prevents the heart from rubbing against the lungs. And that rubbing associated with generation of heat is known as, begins with F and rhymes with miction.
Friction.
Yeah. So the pericardium, because not only is it, as shown here, it just looks flat, but it's actually fluid. It is flat, but it has a layer of liquid pericardial fluid in between the two layers. So it's more like, is often described as like if you take a balloon filled with water and you push your fist into it, your fist will represent the heart, and the balloon filled with water that covers the heart. That's really what the pericardium is all about. Okay. So good. Yeah, so this is fat as shown there. All right. This is just showing then the location of the heart itself, right atria, right ventricle. What does the SVC and IVC stand for? And the PA, anybody?
Superior vena cava.
Correct, superior vena. So if this is the superior, which one this will be down here? Below.
Inferior.
Inferior. And
Inferior.
these, right, and these arteries that actually carry deoxygenated blood, what are they, the left and right? The pulmonary artery. Very good. Very good. Okay. Yes. Oops.
So is that how it looks with all blood in it?
Oh, sorry. Once again, what we're looking at here, this looking more like once again, a cadaver, a preserved specimen. It doesn't, it looks very red. So it looks very much different where in real. That's a preserved specimen. So when it's preserved, it looks like it gets this brownish tinge to it. Let's see if we could have. Yeah, so it looks, so this gives you a more true appearance. You see, it's red, it's tissue, yeah? I know, I don't know if you can appreciate it, from this perspective, but it's through the chest, yeah, retracting his chest there, cavity. But it's very much, yeah, actually into the heart, yeah. It's very much, it doesn't look brown, but it looks red. It looks very healthy in that regard. There's sponging there, right? But the cavity is open there. So that's what it looks like. So this is really, this is a preserved specimen they're looking at from a cadaver. This is not, so it doesn't look like, so if you open your chest, it doesn't look like this, no, to answer your question. That's a very good question. And I should really label these things cadavic specimen as opposed to a live specimen. All right. ECG. You should be able to tell the difference between bradycardia and tachycardia if given a normal beat. How do you tell the difference if it's fast and slow compared to a normal? Let me see. Have a look at these three ECG traces. Normal, fast. What is the difference among them? So tell me first, for the fast heartbeat relative to the normal, what is different? Something measurable. What could you see that is different between the normal and the fast? The fast has something that a normal one doesn't have. What is it? Something
Waves,
that, the waves, how many waves? Yeah, the PQRS, the wave. And how many are the humps you see for the fast?
How the T waves are very close to one another.
They do. And if you're looking at this wave, the trace, the PQRS wave trace, these how many does the fast heartbeat have? 1, 2, 3, 4, 5, 6, 7, 8. 8. And is it 1, 2, 3, 4, 5, 6, 7, 8? Well, it could count this on there. Let's say 9. 9 for argument. Yeah, kind of chunky. 9. And what does the normal beat have? 1, 2, 3, 4, 5, 6. Yeah, so this has more. So in fact, if ever you're given a question, which you're more likely than not will see, let's say you're given an ECG trace like this, and what is the other term when you have a fast heartbeat? There's a term for it. It begins with T.
Tachycardia.
Thank you very much. Yeah, tachycardia. And the slow, now we already established this is how much? 6. 1, 2, 3, 4, yeah, 6. The slow heartbeat has how many of those the PQRS humps? How much does the slow have?
4.
4. Thank you very much. Yeah, 4, you're spot on, right? So this one has 4, whereas this one 9 and this one 6. So that's why this is the fast heartbeat and this is the slow. And when we speak about the slow, there's a word associated with it, begins with B. Not tachycardia, but
Bradycardia.
Thank you very much, bradycardia, right? That's the slow heartbeat. You will take note of this. You will see this again with a high degree of probability, all right? So let's say it asks you which one of these traces is showing bradycardia, which one is showing tachycardia. I could also ask for irregular. Irregular will be the odd man out. right, in this case. Now, why do you think, relative to the normal, why do you think they call this an irregular heartbeat? What is it about it that is irregular? Could you make a comment on that?
The space.
Thank you very
between
much.
the
Very
teeth.
good. Yeah. The spaces, if you notice. So this one is close and you could actually count them because it has these graph squares. So this one is like 1, 2, 3. Whereas between these two, it's 1, 2, 3, 4, 5. Then between here and here is 1, 1, 2, 3. Then these two, it's 1, 2, 3, 4. And 1, 2, 3, 4. And so it's irregular. 3, 5, 4, 4, 5. It goes irregular. Hence the reason it's irregular heartbeat. But just eyeballing it, if you step back and have a look, you see small space, big space, medium, medium, medium. And that's why it's an irregular. Take note of this. High probability you'll see it again. Okay? Right. That would be it in terms of with the heartbeat. Let's go to the next one now. So we look at blood, heart and blood vessels, respiratory system. Let's have a look at that one. And then the last one we'll be looking at is the digestive. Everybody okay so far? Everybody okay?
Yes, sir. Yes, sir.
How the COVID treating you?
Just gotta be safe.
Gotta be safe, that is true. Yeah, COVID it's special indeed. How are you managing with having, for those of us who have children at home or everybody fighting, not fighting, but multi-users on the computer, so you had to ration the time. How is that working for you? How it is, you're missing custard, you're missing seeing your colleagues and them? Okay, I'm guessing you're loving the isolation. I am mad at you because some of them really, you could say firebomb them. There's only cause distress. So better to be alone than to be in bad company, as they say.
No,
Let's
I'm
go.
frightened right now.
Huh?
I'm scared I gotta go back to work tomorrow.
Really?
Yeah.
I hear you.
Well, back towards tomorrow.
I understand. Yeah. Yeah, I understand the trepidation. But if it's one advice I could give you is, something pretty straightforward, notwithstanding all the other protocols that have, nothing could beat wash your hands. For any reason at all, wash your hand. You're standing up, wait, no, let me just go and wash my hands. But just keep washing your hands because we don't realize how much things we touch all inadvertently. So one of the better means of limiting spread of anything, of even viral spread to yourself and to others, is by washing your hands. I cannot stress that enough. So in your going to work tomorrow, try to wash your hands as much, or if you do have the little hand sanitizer, the alcohol sanitizer, just keep, for no reason. So it's not like you touch anything consciously that you remember, no, but just keep washing your hands and you'll be good. You'll be good. Let's go forward. So let's look at 13 and then 14. Okay, we're almost there. We're almost through. It had rained this morning or anybody had rain by them?
Yes, sir. It was the reason I woke up late.
Oh, yeah, it had, yeah, boy.
Last night, sir.
Last night had rain too? What you saying?
From 10.
You're making
from 10
joke?
to this morning, yeah.
You're making joke? What you say? Oh, that mean it had anybody flood? It had flood by anybody? Lord.
Oh, not by me. It wasn't that bad.
Anybody flood? Flood out by them? Oh, Lord, because if it rain, most definitely. Anybody penal David down there? Caroni, Arima, Twin City, Belmont, Picton. Oh gosh, I know it's have flooding there, boy. Oh, but yeah, it really rained. It rained a lot last night. I down San Fernando by my mother, and it rained. It rained a lot. This morning I was teaching a class as well. That class was from 11 to 2. So around 11, and it started to fall very heavily during class around 11, 11:15. It came down very heavily. Okay, let's go. Oh, it's 13. Come on, I need 12. Okay, we look at 13, 11, 12, 13, 14 at the labs. So for respiratory system, you should be able to label the airway if given, right? From the nose down to the alveolus. All right, so the nasopharynx, the oropharynx, the pharynx, which is the area behind, the larynx, your voice box, your trachea, bronchi, bronchioles, primary, secondary bronchioles, and then of course down to the terminal alveolus down at the base down here. What is this muscle down here? I forget. Main muscle associated with respiration.
Diaphragm.
Diaphragm, thank you very much. Right, so there's the diaphragm. Hello, Jello. Which lung is bigger, your right lung or your left lung?
The left. No, the right one, sir.
Right, I know it was
Because
just
the
tested.
left one are from it for the heart.
Correct. I like how you were trying to test me. I applaud you for it. You was like, let me see if sir know what he's talking about. Let me just show that in there. I applaud you for it. Right, so indeed the left, and as you quite rightly said, it has this bit of a notch that is to accommodate the heart. So that's the reason why it's smaller, right, the left lung. So which one has 3, which one has 2? This one will have 3 lobes, and this one would have 2. You know, in truth and in fact, this one does have, anatomically, it does have 3 lobes, you know, except the second lobe is just so small, it shrink because to accommodate the heart. It just kind of choke up on itself, and it just form a very small structure. It's not shown here. It's really small. So for the most part, is negligible, the size of it. So that's why it says it has two lobes, right? Okay, so you should be able to label the entire pathway related to breathing. This what we're looking at here. This is a preserve. This is not even preserved in formaldehyde. This is a waxy model. So I don't know what preservative they use here. It was a plastic infusion or something. All right. Okay, so you should be able to, let's say, label it from this, the nose, the pharynx is the throat, right? We have the oropharynx, nasopharynx, laryngopharynx, right? And then you have the larynx, it is your voice box, and the epiglottis, right? What does the epiglottis do? Well, it's like a policeman or police person in these times of political correctness. And what it does, it covers up. So when you're talking,
That's the trachea.
yeah, so there's the trachea going down to the lung. Yeah, so the trachea going down to your lungs, and behind it you have the esophagus going down to the stomach. Stomach right in the back there, right? So what this does, it covers up the trachea when you're eating, so food doesn't go down. And of course, it opens up when you're talking, so that the air could go over your larynx, which is down near your voice box, so you can get sound. The air could pass over, back and forth over your larynx. Right? Good. So the labeling, important, yes. There's the alveoli, yeah. No. Right, we mentioned that, so that's the main one. So this, again, this is what I mentioned, the larynx, trachea, primary, secondary, tertiary, and then the bronchioles. Right? These are your vocal cord. We wouldn't be asking for you to label this, no. Yeah, pay attention to this one, your alveoli, right? So if given this one, you should be able, the crisscross. Now, note the pulmonary artery and vein, and it's very easy to mix them up. This is the only time it'll be colored red because it has oxygenated blood, the pulmonary vein. All right. And this structure, of course, here, this will be an alveolus. The alveoli, it looks like a bunch of chenet or a bunch of coconuts. All right. Good. That's
So
it.
the same diagram coming back, the same looking diagram coming back.
There's a high probability, yes. So that's why I say, so just pay attention to the one that I do point out. And yeah. Okay. Why did I close that? Yes, so we finished with that one. So the last of the lot now, last one is the digestive system. With this one, you're gonna pay attention to the villi. Let's have a look, see. right? So this is the last one. So the digestive system should be able to label it from mouth to anus all the way down, right? So the liver, notice how big a liver is. Gallbladder. Gallbladder is associated with what digestion of what type of food? Carbohydrates, fats, or proteins? Gallbladder. Well, gallbladder, what is it? It stores what, the gallbladder?
Bile.
Bile, right? And what does bile do to fats? It affects fats, really. Class of A. You hear often that you hear bile emulsifies fat. And what does emulsification of fats look like?
Is it breaking down?
Yeah, it spreads it out. And I like to give the analogy, you know, when you finish cook your stew chicken on Sunday, and you know the pot, you're washing out the pot and you put it in the sink, you fill it up with water. You know, you see like a film of oil that forms on top of the water. Yeah?
Yes.
Right. If you take detergent and you squeeze it into the sink, what happens to the layer? You see how it breaks up? You know, you see it pulling away from each other into, you know, and it form like little, like bigger circles. You ever notice?
Yeah.
Right. That's what emulsification is. It just means breaking up of fats. And that's what it does. That's what bile does. It's very important for that. Now, in terms of the movement of fats, because now fats are big, right, in terms of their structure, they're relatively big, as we spoke to. So therefore, where do they pass? Do they pass through lacteals or do they pass through capillaries in the body? So your lacteals is part, of course, of your lymphatic system. Which one do they pass through? Well, if you had a choice to make, where do you think they'll go? First of all, let me ask this logically. Now, fats are large. So if you're large, which one you'd want to go through, a large area or a small area?
Large one.
Right. So the question now is, which one is larger, lacteals or capillaries? And the answer is not capillaries, but
Lacteals.
yeah.
I'm trying to
Go
figure
ahead.
out in my brain what the lacteals look like.
Sure, let's have a look. So that is, well, first of all, to understand, I hope you can appreciate why it is the body, the machine, why it uses the lacteals. For that very simple reason that they are bigger. Fats are big structurally. Let's have a look to see what fats look like. Just appreciate it. You can understand that, yeah? So fats, by their very nature, they're big and bulky, right? So for instance, these are triglycerides, but they're big. These are showing the molecular representation of the carbon and hydrogen atoms that make it up. So if you notice here, you know, there's a fat, look, it's big, right? So lots of carbon, yeah, carbon backbone with hydrogens. Here you have an aldehyde group, C double bond O. Well, this is a modified acidic group, C double bond O, right? And then it's now attached to another one. And so it's big, it's relatively big. So the question is, as you quite rightly ask, what is the width of the lymphatic? Right, from 10 to 80 micrometers in diameter, right? So they could go up to, they're a little wider than capillaries. Now capillaries, they're more fixed in length. They're about 8 to 10 micrometers, right? So whereas the capillary is about 10, the lacteals could go up to 80, almost 8 times their size. So they're bigger. So we can appreciate that fact first, yeah? The lacteals, they're bigger, 8 times the size of the thing. Now let's have a look at let's see what it looks like, a lymphatic. So the lymphatic system, right, it runs through, this is where all over your body, and this is where lymph fluid or tissue fluid actually flows through. So this is fluid less And this is really, when you look at the lymphatic system, this is where the circulation of the white blood cells actually occur. So they move through here, then they enter the general circulation, and now they could penetrate the walls of the capillaries and get to the site of infection, let's say, right? So if we were to look, this is we're looking at the lumen of the small intestine, and you have, there's a lymphatic vessel that passes at the base, and it runs up the, these are the villi. So throughout the lining, when you look at the lining of the small intestine, they're crisscrossed with these villi. And in the middle of the villi, you have these lacteals. So what happens is, yes, you eat your food, yum, 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 right? You have diffusion across the membrane here into the blood circulation. And running at the center, so you have a capillary bed occurring right at this point between, so these are, here it is, you have an artery and a vein, you have a venule, which is a smaller representation of a vein, and the arteriole, smaller representation of the artery, and then you'll have a capillary bed, and in the middle of the capillary, so the capillary bed runs between the arterioles and the venules, and at the center you'll have the lacteal, right? As we mentioned, the lacteal is about, in terms of the width, is about 8 times the size of a capillary, so that when absorption occurs, there are nutrients, so remember the food is running on the outside, so when it is, fat is encountered, fats are too big to pass through the capillary bed or to enter into capillary. So what they do instead is they enter into the lymphatic system. So the fats will enter here and then they travel through the lymphatic artery. And that's, your body is so well designed, it recognizes that, look, if I let those fats go through the capillaries, will cause clogs. So let me send them through instead, the lacteals, and that's how they pass through. So that's how you have the lymphatic system is associated with the absorption of fat. And the fat, this is done via the lacteals. And the lacteal is nothing more than an extension of the lymphatic vessels that run inside of the villus, which is located in the small intestine. We good? Guys, okay?
Yes, sir.
Okay, good. All right, let's continue. Right, so let's go ahead. This is, now we wouldn't concentrate too much on that one. That's the stomach. Of course, when you swallow, it goes down by peristalsis, down to the level of the stomach. Your tooth, pay attention to this, you should be able to label a tooth, tell the major parts. So all of these labels are important on the tooth, the crown and the root, and the different other portions of it. The stomach, just to take note of the sphincters, every pyloric sphincter. What's the name of this sphincter, you think? I'll give you a hint. What tube leads down to the stomach? What is the tube that leads down to the stomach called? It begins with an E, or it begins with a silent O. So the trachea goes to the lungs. Thus, thank you very much, yeah? So what do you think is the name of this sphincter?
Esophageal.
Thank you, yeah. This is esophageal sphincter, all right? So this is the esophageal sphincter, yes? And this one is the pyloric. You mentioned cardiac sphincter. Is there a cardiac sphincter? It sounds familiar. Let's have a quick look and see. Yeah, good point. Cardiac sphincter. Between the esophagus and the stomach, which is the alternate name for the esophageal sphincter, also known as the cardiac sphincter, right? So you were spot on there. So one is more related to the anatomy associated with the tube that's coming down. The other one related to the area, because right behind it, the stomach, stomach, superior to the stomach, you would get the heart. So that's why they call it the cardiac sphincter. Let's see its position here. So yeah, here's the stomach, which is actually is inferior. Yeah, it's inferior because the heart is up here, inferior to the, so the heart is up here, really right here. And so inferior to it, you have this one. So this is the cardiac or the esophageal sphincter. What this is showing here, regurgitation of food, right? Some people have it, and it's called, you get this burn at night. And what is that known as? Heartburn, also known as acid reflux disease. Yeah. So some people have it. What you could do, of course, to alleviate it sometimes is just, so nothing more than putting pillows under the bed to prop it up. Yeah, good. Right here it is, you have the alveolar. Here it is the villi, right, showing the villi. With the villi, just take note, this diagram here should be able to label it. All right, so take note of this one. No, not these, nope. This one, if you're given it, you should be able to identify what it is, where it's from. You know, so if it has given this, where is this from? You should be able to say, well, that's from the pancreas. And the absorption, small and large intestine, no. Nope. Yeah, the parts of the large intestine, ascending, transverse, descending, sigmoid, colon, sigmoid colon, sigmoid, rectum, and anus. And that's it. All right, so what did we do today? Well, we had a bit of a revision session in terms of, we had a revision session in which we looked at the things necessary for successfully getting a good grade for your Sphincter next week. So what did we look at? Well, we looked at the blood, mentioned, the different things that we should look at, should look for. And again, in our revision, we should put emphasis on diagrams, images. That's where the majority of the work will be coming from as it relates to the spotter. All right, we looked at the heart. We mentioned not too much on the blood vessels, the nomenclature or the naming of the blood vessels. No, all right, but we did point out some things which are important relating to the heart. The respiratory system again as well, we did mention certain things. And I think we all learned something as it relates to the lacteals, which is a part of the lymphatic system, very important for the transfer of lipids in the body itself. And the reason why, because they're larger than the capillary bed, and you run the risk then of getting a clot, of having a blockage of the capillaries if you take up the lipids in them, right? And that's it. Any questions? Nope. All right, let me just finish roll and I will have you on your way.